spectroscopy to characterize the pathological polymer of alpha-1 antitrypsin. Alpha-1 antitrypsin is a 52 kilodalton glycoprotein. It circulates the plasma, and its role is to regulate the proteolytic effects of neutrophil elastase in the lungs during inflammatory events. It's a metastable protein, so it's able to undergo large conformational changes, and that helps with its function. So what it does is the protease will bind to this pink loop called the reactive center loop, and it will flip it to the opposite pole, and that inactivates the protease and causes the reactive center loop to insert into the central beta sheet as a 6th strand. This metastability can also be its downfall, as you get spontaneous insertion of that reactive center loop that has pathological consequences, which I'll bring up in a second. So we've been able to look at the monomer forms of antitrypsin through NMR spectroscopy, and what we're showing here is the wild type that we refer to as M, and one of the disease-associated mutants that we call Z. So this is all completely natural abundant spectrum, purified from the plasma of healthy and antitrypsin deficient patients. So that means we're not doing any bacterial expressions with enhanced isotopic labeling of NMR-active nuclei. That means we're having to do very long collection times. Each of these took about 80 hours each at high magnetic fields. And this is really exciting because when we were comparing M and Z crystal structures, we weren't able to pick up any distinct structural differences, whereas when we look at it by NMR, it's sensitive enough to local chemical environments, and we're able to plot those structural differences between the two variants. Something that we haven't been able to characterize yet is much about the polymers. So the mutant forms will cause that reactive center loop to insert, and then you can get polymer chain formation that will aggregate within the liver of patients, and subsequent lack of working antitrypsin in the plasma. So these polymers seem to be as beads as a string. This is a negative EM image taken from the liver tissue of antitrypsin deficient patients. We've been able to reconstruct the general molecular shape of them, but crucially what we're missing is actually that intermolecular linkage and the polymer structure. So these are so difficult to characterize because they're highly flexible. We're not able to use crystallography, and cryo-EM is incredibly difficult. So we're using NMR spectroscopy that can look at dynamics in solution instead. So we started off this project with taking monomer antitrypsin that's been isotopically labeled. We put it in the magnet at high temperatures to induce polymerization by heat perturbation. And what you're seeing here is a time course of, at time point 0, you've got the high intensity peaks will be the monomer, and then those will decrease over time as the protein is polymerizing. Through some further purification, we were able to get some high resolution spectra of these polymers and confirm them by negative stain EM that they are in fact these circularly permuted chains. So something in the back of our mind during this project is another form of antitrypsin that we've been working with, which is called cleaved antitrypsin. That's where the native structure has been clipped at the reactive center loop with an endoprotease, and that causes that reactive center loop to insert into the beta sheet as a 6th strand. It's been a question of if the cleaved and the polymers are structurally equivalent, and so when we overlay the spectra, you can see that they do overlay extensively, and that's telling us that the local chemical environments of a cleaved monomer is in fact very structurally equivalent to a multi-subunit polymer. And just to hone in on the sensitivity of this technique, it works actually to cleave at a slightly different point of that reactive center loop, causing a slightly longer loose loop and a shorter reactive, shorter inserted strand. We can actually pick up the chemical shift perturbations that are happening from such a slightly different structural change. And the reason we're so interested in cleaved antitrypsin is that it is a bit more characterized than what we have for our polymers, so we can use what we know about the cleaved structure to inform us on the polymer structure. We also have a toolkit of monoclonal antibodies. One of them is a polymer-specific one, and it can recognize polymers derived from the liver of patients, anything that's been heat perturbated, and cleaved antitrypsin. So given it's had this shared epitope of cleaved antitrypsin and the polymers, it's been a hypothesis that there must be some structural similarities between the cleaved and the polymers, but this is the first actual confirmation we've had of that. The cleaved state is also particularly interesting because it mimics a single subunit of a polymer that's undergoing one of the prominent polymerization models in the field. There's a number of polymerization models. The one we happen to lean towards is the C-terminal model. That's where it's proposed the C-terminus of one antitrypsin molecule will insert into the body of next, and that's how you get those intermolecular linkages. So the fact that we have the cleaved state that's representing one single subunit of a polymer that's undergoing the C-terminal mechanism, and we've seen that it is structurally equivalent to our heat polymers. That tells us that these heat polymers are in fact undergoing the C-terminal mechanism of polymerization. So this is always recombinant samples that's been artificially induced, and so next we wanted to think about the actual polymers that are derived from the livers of our patients. So what we do is we get transplant patients that have antitrypsin deficiency and severe liver cirrhosis. We go through an extensive purification method and isolate these polymers by chain size, so going from largest to smallest. I take the two smallest polymer chains I can, and we use 1D spectroscopy. So this is all human-derived material, which means it's incredibly dirty, it's natural abundance, it's very low yield, and they're high molecular weight. So rather than getting those nice high-resolution 2D spectra that we could get of the recombinant material, we have to settle for just looking at the high-field methyl resonances of 1D proton spectra. And I still think this is a very powerful fingerprinting technique when making comparisons between different samples. So if we have the recombinant wild-type monomer compared to monomer that's taken from antitrypsin deficient patients, it obviously has very different spectral profile. And then if that same material has been cleaved so that it has a short inserted strand, it again has differences that are distinct from something that has the full inserted strand. So based off of these, we can look at our liver samples. And what's important to note is that the liver samples have nearly identical resonances between themselves, telling us that regardless of how many subunits are in these liver polymers, they're identical structures. And they closely overlap and overlay with the resonances of our plasma cleaved sample, just as we saw with our heat polymers in vitro. So given the extensive overlay, we can transfer the assignments we have from the cleaved and to our liver spectrum, and only a few could be reliably assigned. However, we're quite lucky in that these are quite well distributed throughout the whole antitrypsin molecule. And NMR doesn't just report on single amino acids, they're reporting on that local chemical environment. So if we're just being conservative and looking at a 2 angstrom radius of our probes that we'd be able to pick out, we can see that the contacts that are extensively similar to our cleaved sample do cover a wide range of our structure. And they also cover points which are quite crucial to what we're looking at, which is the insertion of that reactive center loop into the beta sheet, and right behind the where that C-terminus is being inserted. So I hope I've been able to show you that our heat-induced polymers and our liver polymers are very structurally identical to our cleaved samples, our cleaved monomer, and due to that fact, both are very likely to be undergoing that C-terminal mechanism of polymerization. Obviously, it would be nicer to get a 2D spectrum out of this, so something that we've started working on, and actually I was very lucky to work with Instruct-ERIC and Marcus at Utrecht with some solid state. And I've continued a collaboration with Dr. Trent Frames at Warwick University. We've taken isotopically labeled heat-perturbated polymers, put them into a magnet, and defined our spectral resolution. And more critically, we've started working with the rawest material possibly for our liver polymer. This is pure inclusion body pellets of our antitrypsin. We've washed it with detergents to help remove lipids, and then packed into a rotor and see if we could get any signal. We're quite lucky in that we did in fact get carbon-13 signal, which we did not quite expect, to be honest. And we're still working with this currently to further optimize it and be able to make spectral comparisons with our recombinant material as we did with the solution state. I'd like to thank my supervisors, David Lomas, James Irving, and my collaborators, Chris Wardby with the solution state NMR and Trent Franks with the solid state NMR, and all of you for listening. Thank you.